All right, and our last speaker of this section is Erin O'Neill, who is an attending physician at CHOP, specializing in inherited retinal diseases, and also a K-12 scholar who's going to be telling us about an exciting new project on the gut microbiome in pediatric retinal degenerations.
Thank you so much. Thank you for the opportunity to speak. Since joining the K-12 program last summer, I've been very fortunate not only to expand the work that my mentors have talked at length about in terms of expanding gene therapy trials to kids, as well as developing new measures of visual function in young kids with severe vision loss, but also to explore a topic that I've been very passionate about for many years. And I'm delighted to have the opportunity to share our progress so far in evaluating the intestinal microbiome in pediatric patients. So I wanted to take a step into the clinic so you guys can understand what my day-to-day is like and understand what motivates me in my research. I recently met a 6-year-old boy who was having a bit of trouble seeing the board in school, went to Warby Parker for an eyeglasses exam, and was found to have an abnormal retina. He has no past medical or ocular history, but there's a very strong family history of early onset vision loss, particularly in male members of the maternal side of his family. His maternal uncle is legally blind in his 50s. Multiple of mom's male cousins and the maternal great-grandfather also had early vision loss as well. On my exam, his visual acuity was 20/25 in the right, 20/20 in the left, and the color vision was intact on FD15. Base exam was pretty unremarkable, as was his refraction, but when we did his DFE, and I apologize, I know one of these is a right eye and one of them is a left eye, but when working with kids, we kind of just take the best that we can get. So you can see that he has vascular attenuation. I'm gonna try this out, I don't know if I can do it. Yes, he has vascular attenuation, and he has peripheral white dots. And then when you look at his fundus autofluorescence, he has an obvious ring of hyperautofluorescence right here along the arcades, and then hypoautofluorescence peripherally, where the area of the white dots is. On the OCT, you can see he has preservation of the ONL and the photoreceptors at the fovea, but loss of those as you get to further eccentricities. The next step for him was genetic testing, which unsurprisingly came back with a pathogenic mutation in RPGR, leading to a diagnosis of X-linked RP. And at this point, the parents asked me the same two heartbreaking questions that every parent asks me in every single visit that I have. Is there anything we can do? For most of them, and as this family in particular is very aware, they are just waiting for their beautiful and vibrant child to become legally blind in young adulthood. And while in his case, there may be the opportunity to participate in an upcoming clinical trial, we're all aware about the recent disappointing results for RPGR gene therapy, and so it's not very heartening when we're trying to have this conversation. And every family also asks me, what else can we do? Is there any lifestyle modification we can make to help preserve the vision that our child has? There's been a number of supplements that have been tried in RP, but recently the AAO put out a report in which a number of these supplements were analyzed, and they found no convincing benefit from any of the commonly tried supplements, including lutein and vitamin A. And as most of you may be aware, there is an ongoing clinical trial called NAC-ATTACK of oral N-acetylcysteine for RP, the results of which we are awaiting hopefully within the next couple of years. So I have been wondering for a long time, is there anything else that we can do to help these kids? And for those of you who knew me back in the pandemic, you'll remember that when everyone was cultivating their sourdough starter and making whipped coffees, I became really obsessed with the intestinal microbiome. And if anyone's not familiar with that term, it basically refers to the bugs that live in our gut, as well as their genetic material. They actually outnumber us by a ratio of 10-1 in terms of the number of cells, and they contribute 8 million genes to make us who we are as compared to the only 20,000 that we contribute of our own. There's over 1,000 species that have already been identified, and while some sites, like the intestine, have a staggering size, other sites, like the ocular surface, and until recently we thought the retina, are paucibacterial. The intestinal microbiome has been linked to a number of systemic diseases, including neurobehavioral diseases, asthma, cardiovascular disease, a number of cancers, metabolic syndrome, and IBD. In terms of the eye, it has been evaluated in terms of dry eye, uveitis, and perhaps best explored age-related macular degeneration. So at this point, you're probably wondering, Erin, how did the bacteria that live in our gut actually mediate disease at distant sites in the body? We do know that some bacteria that live in the gut can promote differentiation of inflammatory Th17 cells there in the gut, while other perhaps more beneficial bacteria can promote differentiation of regulatory T cells, as well as the short-chain fatty acids and other metabolites produced by those bacteria. And we know that the overall balance in our body of regulatory to inflammatory T cells being unfavorable can result in inflammatory systemic disease. Some of these microbial peptides can also induce antigenic mimicry, leading the body to believe there's a foreign invader rather than a commensal organism living in the gut. And finally, we know that the disrupted intestinal barrier can result in spillage of nonspecific bacteria and their antigens into our lymphatic and our bloodstream. There has been some preliminary study to investigate the role of the microbiome in inherited retinal degeneration. So a group at the University of Alicante looked at a mouse model of RP, the RD10 mice, and found not only a decreased overall species diversity in the gut microbiome of those mice as compared to healthy controls, but also a complete absence of 4 common gut microbiome species, Prevotalaceae, Rickenella, Myribaculaceae, and Bacilli, as well as an enrichment of Bacteroides species in the gut of the mice. They then went on to publish work on a group of healthy control mice fed a normal diet in the dark red or a high-fat diet in the light red to the diseased mice fed a normal diet in dark blue or a high-fat diet in the light blue. And they found both decreased ERG amplitudes and spatial frequencies in the diseased mice on a high-fat diet, suggesting that we maybe are in fact what we eat, and there might be a dietary component to progression of retinal degeneration. Now, I want to take a brief interlude because this is going to come back later to talk about a very different retinal disease that affects children. Retinopathy of prematurity affects babies who are born early or who have a complicated postnatal course. It results in abnormal vascular development, can result in hemorrhage, or in the more advanced stages, retinal detachment and permanent vision loss. Dimitris Gondra and Sarah Hilkert Rodriguez published a pilot study in which they looked at a small group of babies who developed type 1 ROP and compared their early gut microbiome to those of babies with similar risk factors who did not develop ROP. They found enrichment of Enterobacteriaceae in the gut of those babies at 28 weeks post-menstrual age, as well as enrichment of arginine and proline metabolism, histidine metabolism, and oxidative phosphorylation in the babies who did not develop ROP. And that same group followed this work on with a poster at APOS this year in which they examined a large cohort of 720 babies, 50 of whom developed type 1 ROP, and found that there was, after accounting for all the common covariates in ROP, gestational age, birth weight, BPD, NEC, sepsis, there was an increased risk of developing ROP in the babies who were treated with beta-lactam antibiotics. And as any of you who have ever taken antibiotics know, that can definitely induce gut dysbiosis, and so there's a possible pathogenic mechanism there that we need to explore further. So with all of this preliminary study in mind, we set out to create a center to evaluate the intestinal microbiome as it relates to ocular diseases. At this point, since starting on the K-12, we do have an IRB-approved protocol to recruit both pediatric and adult patients who have any ocular disease that is known to affect children, or in the case of healthy controls, no ocular disease at all. The patients are sent home with a stool kit in which they send back at their leisure to CHIPS, which is the Center for Human Phenomic Science at CHOP, where they're processed and stored, and we've partnered with the Penn CHOP Microbiome Core to perform the sequencing and bioinformatics analysis. Our initial targets as part of this study include inherited retinal disease, which is obviously my area of interest, and we're looking specifically at patients with CRB1 and RPGR X-linked RP, as well as ROP. We're finalizing our protocol to investigate non-infectious uveitis, particularly related to JIA in pediatric patients, and we're certainly looking forward to partnering with others on future targets. For anyone who needs just a brief history on CRB1, it's a relatively common cause of LCA or RP. It classically presents with a nummular pigmentation of the retina. There's laminar disorganization, early macular involvement, frequently with CME, and there's classically para-arteriolar RPE preservation, as you can see in this patient who is in fact enrolled in our study. Go back. The gene product localizes to the photoreceptor inner segments, the Mueller cells, and the epithelial cells. And we recently have learned it's not only involved in retinal development, but also in formation and maintenance of adherence junctions, not only in the retina, but also in the gut. So in 2024, Peng et al. Published this pivotal work in which they found that loss of CRB1 in a mouse model of disease resulted not only in loss of the enteric adherent junctions, but also the apical junctions of the RPE. This resulted in direct translocation of bacteria from the intestine into the retina, which was observable via direct visualization. They also found enriched pathways for bacterial identification and inflammation in the inferior retinas of these mice, which is where we see early signs of retinal degeneration, as well as microglial inflammation and enrichment of 5 specific bacterial species. They also found an enrichment of Akkermansia in the gut of the mice, which was not observed as a high abundance in the wild-type mice. Raising these mice in a germ-free environment resulted in less microglial inflammation and preservation of the retinal structure. Also, prenatal and postnatal treatment with antibiotics resulted in the same preservation of the retinal structure in these mice. Finally, they also restored CRB1 expression via AAV2-mediated gene delivery to the intestine, but not the retina, and found that not only did that restore the enteric adherens junctions, but also resulted in amelioration of the retinal phenotype. As I mentioned, we're also looking at RPGR X-linked RP due to the well-known and common association of CME in patients with RP. And in fact, up to 50% of patients will demonstrate a pseudo-inflammatory CME at some point in their disease course. The proposed mechanisms I have listed on this slide, including possible breakdown of the blood-retina barrier, dysfunction of the pumping mechanism in the RPE, Mueller cell dysfunction, anti-retinal antibodies, or vitreous traction. And while it's most commonly observed in patients who have autosomal dominant RP, we don't see as much of that at CHOP because of the average age of diagnosis. We do see a lot of kids who have RPGR X-linked RP, and at the time I prepared this slide, 1 in 5 of them had CME, whereas the remainder did not. When these patients have CME, I have the typical therapeutic pathway that I follow, starting with topical carborganic anhydrase inhibitors and then escalating to orals or topical ketorolac if it's a young child. Unfortunately, this isn't always successful, and almost universally my patients tell me that even if I'm able to restore the normal retina structure, they don't really appreciate any improvement in their vision from it. So I wanted to identify possibly an alternate therapeutic pathway for this inflammation. So for our IRD arm, we're currently recruiting patients with CRB1 or RPGR, both at CHOP and UPenn. They have a single visit in which we collect a medical, surgical, and ocular history, as well as a detailed dietary history and an antibiotic history. Patients have to have been off of systemic antibiotics for 2 months in order to participate and send in their stool sample. They don't have to come in for a separate visit for their ocular phenotyping. We simply do that via chart review. And then, as I mentioned, they send their samples into chips where they're currently being processed and stored until we have enough to fill a tray, do the economics of actually doing the sequencing of this, at which point the CHOP microbiome core will sequence and analyze it for us. As of today, we've recruited 6 patients with CRB1, 5 patients with RPGR, and 2 controls who are both siblings of patients in the other 2 arms. In terms of the current status of our CRB1 patients, I have them listed here. They range in ages from 3 to 26. Not all of them were initially diagnosed with CRB1. It can be a bit of a masquerader. Our most severely affected is just 3. His visual acuity that we can measure is light averse only. And then our best preserved patient has visual acuity of 20/40. You can see that 3 out of them have obvious CME, and then the 2 oldest patients with the most severe degeneration don't have any CME, but we know at least one of these has had it in the past, likely prior to her complete structural loss at the center of her retina. In terms of our RPGR patients, they range in age from 6 to 18. Their visual acuity is relatively preserved as we would expect based on the natural history of the disease. One of them you can see has obvious CME at this point. The oldest patient has microcystic edema, and then the patient who is in P4 has had CME in the past, but is currently controlled on oral acetazolamide. So our next step, I'll go back so you guys don't get distracted. Our next step is to complete recruitment of both of these arms as well as recruit additional healthy controls so we can sequence their results. The design for the ROP arm of the study is a little bit different because we have the benefit of a large cohort of patients who were recruited and treated at the Penn NICU. There's 166 babies in their cohort, 117 of whom have already had their stool samples analyzed. These babies gave weekly stool samples from birth to discharge from the NICU, which for one of them was all the way at day 108. And so between the 117 who are sequenced, we have over 900 samples, which are currently in analysis at the bioinformatics core. We do have ROP data on a subset of them. 58 of them did not develop ROP, 36 had non-severe ROP, and 8 had ROP such severe that it required treatment. All of them received laser, and 2 additionally received Avastin. Thanks to the hard work of our collaborators at the Penn NICU, we already have the data for the common covariates, which we would want to look at in an ROP study, include gestational age, birth weight, mode of delivery, presence of prenatal or postnatal infection, as well as in a subset of them, serum IGF-1 and VEGF levels, which is obviously of interest when we're examining ROP. The next step is to, as I mentioned, analyze their intestinal microbiome composition, which we will do weekly from birth until development of ROP or severe ROP, or until 40 weeks post-menstrual age. I've already gone over some of the next steps that we have. We are also hoping to expand collaboration both within CHOP and UPenn, as well as with our colleagues at the University of Alicante, who I mentioned earlier, who had done the work on the RD10 mice, but also recently presented their results from a large cohort of patients with central areolar choroidal dystrophy due to mutations in the PRPH2 gene. So I wanted to thank my mentors on my K-12 who are on the top row, as well as my other collaborators at CHOP who are on the bottom row. Thank you so much.
Great talk. Questions for Erin?
Thanks, Erin, for something different for us and very interesting. What are your thoughts on, do you think it's differences in the microbiome that are inherent due to somebody's genetics, or do you think it's purely dietary as what you might find in the differences between patients?
Yes, I don't know that anyone has really explored the genetics of microbiome. Certainly, there's been a lot of work done in terms of our immediate postnatal course, how we were delivered, how we were fed, what kind of environment we were in the early postnatal course. But then once we become older, as such, we know mostly based on our diet. I didn't really put a slide up there about the kids who are enrolled, but for what it's worth, the kids we've enrolled so far all eat a full and varied diet. There's one vegetarian, but all of the other kids eat fruits, vegetables, refined sugars and grains, dairy, all of the things that we think may influence the gut microbiome. They all eat a relatively similar amount of that. So I think more to be done in terms of the genetics of it.
Great.
If kids have to go on antibiotics for any reason, ear infections, things, does the microbiome, does it eventually go back and revert to the original after a couple of weeks, or do you have to, do anything with those patients if they're already enrolled?
Yeah, so based on my conversation with the real specialists in this in the GI department, as long as they haven't been on antibiotics for 2 months prior to enrolling, we're fine. It goes back to where it was. They only at this point provide one stool sample. So we have one patient who enrolled but was on antibiotics at the time of enrollment. So they got sent home with their stool sample kit and then they sent it back in after it had been 2 months.
Great talk, Erin. So the Penn Biobank is getting genomic sequence on patients who enroll. I imagine that CHOP has a similar thing, so you might actually be able to answer Ken's question about relationship between genetics and microbiome.
Yeah, one of my patients said she had just enrolled in and she was like, oh, you don't have to take blood? Great, I'll take that stool kit home, no problem. So yeah, I think it would be nice to collaborate with them.
Great. And also, once you discover which microbiome is associated with better or worse outcome, what kind of interventions would you envision?
So a lot of people take probiotics. What I've heard lately is that they are of questionable efficacy, and that something like a fecal microbiome transplant, which sounds a lot scarier than it actually is, or possibly an antibiotic treatment, for example, might be more efficacious. But we're a little too early stage for that.
So I read a paper that the microbiome of babies born vaginally or by C-section is different, and I would think that a lot of your babies that are with ROP are C-sections. Is that true or not true? And so would you see differences within those two populations?
Gil is definitely the ROP expert in our group, so I don't want to answer that confidently, but I know that there are certainly babies who are born vaginally who develop ROP, and it certainly is not entirely C-section that's driving it. We do have that data in our cohort. We just have not analyzed that yet. But yeah, it certainly is different.
All right, I think Erin's talk on the gut microbiome was a perfect segue into lunch. And so I want to thank all the speakers for really fantastic talks today and also for staying on time. Everyone gets a gold star for that.